Hello everyone, human reproduction, subtopic oogenesis. Let's study about the oogenesis. You know that oogenesis is the process of formation of the female gamete, that is the ovum, right? So here, let's say the structure of the ovary. You know that ovary is covered with the thin epithelium. So just imagine this is the ovary and that is covered by the epithelium, and the inner space that is called as ovarian stroma. And here the ovary is divided into two parts, the outer one called as cortex and the inner region that is called as medulla. Then yes, this is the structure of the ovary. So here you can see the medulla region as well as the cortex region. So mainly in the cortex region, the development of the follicles and the Graafian follicle, so all the oogenesis events that takes place in the mainly in the cortex region of the ovary. Yes, here the oogenesis is the process of formation of the mature female gamete. You know that in the spermatogenesis that is initiated during the puberty, right? But the oogenesis that is initiated during the embryonic development stage, when the couple of million gamete mother cells, oogonia, are formed within the fetal ovary. So when the female baby in the mother womb, so that is called as embryonic stage. So at the embryonic stage only the oogenesis begins. Means millions of the gamete mother cells, oogonial cells, so that is formed within the fetal ovary, and no more oogonial cells are formed or added after the birth. All the oogonial cells are formed before the birth of the female baby. Okay, so no more oogonia is added after the birth, or no, oogonia is not formed or added after the birth. So oogonia cells start the division, so and enter into the prophase 1 of the meiotic division and get temporarily arrested at the stage is called primary oocyte. So here the oogonial cells which are, that start the meiotic cell division, the oogonial cells that all start the meiotic cell division, but that are arrested at the prophase 1 of the meiotic cell division. Okay, so that is the first step. So each primary oocyte, so that is uh, called as primary oocyte, oogonial cell that start the division, so that will become primary oocyte. The primary oocyte that is arrested at the prophase 1. So here the primary oocyte that start or that gets surrounded by the layer of granulosa cells and called as primary follicles. So here this is the primary oocyte. So this primary oocyte that starts the uh, some layer of cells that start form around the primary oocyte, that is called as the primary follicle. Okay, a large number of these follicles degenerate during the phase of birth to the puberty. So this large number of these primary follicles that degenerate. So at the puberty, only 60,000 to 80,000 primary follicles are remaining. So at the time of puberty, only 60 to 80,000 primary follicles are remaining. So this is called as primary follicle. Then the primary follicles get surrounded by more layers of granulosa cells, a new theca, and are called secondary follicles. So here see the difference between the primary follicles and the secondary follicles. So these are the secondary follicles. So the more layer of granulosa cells, so that start around the primary follicles, that is called as secondary follicles. Okay, then the theca layer Okay, the secondary follicle soon transform into tertiary follicle, and which is characterized by the fluid filled cavity antrum. So here you can see the secondary follicle that transform into tertiary follicle. So inside the tertiary follicle, primary oocytes are present, right? So here you can see the characteristic cavity formation inside the tertiary follicle, that is called as antrum. That cavity is called as antrum. So that is called as tertiary follicle. The theca layer is organized into inner theca interna and outer theca externa. So inside the tertiary follicle, primary oocyte is present, right? So the primary oocyte within the tertiary follicles grows in size and completes its first meiotic division. So when it completes its first meiotic division during the puberty, okay, it is an unequal division resulting in the formation of large haploid secondary oocyte and tiny first polar body. So here the tertiary follicle that contain the primary oocyte, right? So this primary oocyte that is arrested in the prophase 1 stage now completes the first meiotic division and forms the secondary oocyte, okay, and the first tiny polar body. Secondary oocytes retain bulk of nutrient and rich in cytoplasm of the primary oocyte. Okay. Then the tertiary follicle further changes into the mature follicle or the Graafian follicle. So inside the tertiary follicle, so what is formed? Secondary oocyte is formed, and this tertiary follicle that develops into Graafian follicle, mature Graafian follicle. So here you can see the secondary oocyte and the antrum, and this characteristic arrangement of the granulosa cell layer that is called as the Graafian follicle. Okay, and the Graafian follicle, thus, inside the Graafian follicle, secondary oocyte is there. So that forms the new membrane called zona pellucida and surrounding it. The Graafian follicle ruptures when the Graafian follicle become mature. It ruptures and release of the secondary oocyte takes place. The secondary oocyte released from the Graafian follicle, and that is called as ovulation. Okay, so remember the Graafian follicle that releases the secondary oocytes, so that is not, and that further undergo the meiotic 2 during the fertilization. Okay, so here you can see the schematic representation of the oogenesis. See here, oogonial cells, so that are formed just before the birth of the baby. Okay, so in the fetal life, what happens? The multiplication of the oogonial cells takes place. Oogonial cells are diploid in condition, and the multiplication of the oogonial cells takes place in the fetal life just before the birth. Okay, and these oogonial cells that develop into primary oocyte, so that will become mature, and this primary oocyte that undergo the first meiotic division and arrested at the prophase 1 of the first meiotic division. Okay, so then the birth of the baby takes place. So during the birth to the puberty, the primary oocyte that is arrested in the first meiotic division. Okay, so during this phase, the primary oocyte that develop into primary follicle, secondary follicle, and the tertiary follicle. Okay, inside the tertiary follicle, what happens? So when the puberty, the tertiary follicle, that inside the tertiary follicle, the primary oocyte that completes the first meiotic division and forms the first polar body and the secondary oocyte. So during the puberty, the first meiotic division that is completed by the primary oocyte, and that forms the secondary oocyte and the first polar body. And the secondary oocyte that is present in the Graafian follicle. So the Graafian follicle that contain the secondary oocyte and the characteristic distribution of the cells. When the mature Graafian follicle that releases the secondary oocyte, so the secondary oocyte that enter into the fallopian tube, when the sperm penetrate the secondary oocyte, that complete the second meiotic division and the release of the one ovum, and it forms the one second polar body. Okay, so this is special about the oogenesis. The first thing is no more oogonial cells are formed after the birth. Okay, so during the puberty, what happens? The first on the primary oocyte that completes the first meiotic division and form the secondary oocyte. The secondary oocyte that is released by the Graafian follicle and which reaches the fallopian tube fallopian tube during the fertilization. When the sperm penetrates the secondary oocyte, then the release of the ovum, which the secondary oocyte that completes the second meiotic division and the release of the ovum takes place. Okay, so this is about the oogenesis, the schematic representation and the different steps involved in the oogenesis. Thank you.